I appreciate people coming today. This is the September 7th meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. As always, I need to remind you that the meeting is live streamed and recorded for those who are unable to attend in real time. And I think it's a particularly exciting talk today, so I'm glad that people are able to come in person, and I appreciate our speaker being willing to interrupt class duties to do this presentation. So we're very appreciative of that. As always, Tom Helikar and I are available to take your suggestions and critiques. Jim Sluka and Lorenzo as well. Please feel free to contact us if you have any concerns or questions or ideas about how to make these presentations more effective. We have our usual communication channels. They work better if you use them. We have that new GLIMPRINT.org website as well. Please help us populate the IMAG wiki page and the GLIMPRINT page. We very much appreciate everything you can do for that. We have our growing archive of videos, which we would love to have people help with. Let people know what they are available there. And we have a LinkedIn page and also a Facebook page and a Twitter account as well. We have, again, the Global Alliance for Immune Prediction and Intervention page. Very much appreciate your help with letting people know about that. And we have that MSM Viral Twitter handle, which you could use also to help us get the word out. There are a couple of announcements that we want to make. There are some new RFAs that might be relevant to this community. There's NSF 23616, Ecology and Evolution of Infectious Diseases. This includes testing mathematically computed models that elucidate infectious disease systems. They're expecting to have a number of large-scale awards, and these could be quite interesting. Includes pathogen transmission dynamics, which of course is one of the things that today's lecture will get near to, I hope. And so that might be of interest to many people in our community. There's also an NSF 23614, Smart Health and Biomedical Research in the Era of Artificial Intelligence and Advanced Data Science. This one also might be something that could be well connected to the topics of interest to our community. As always, if people are aware of new RFAs or have projects or grant opportunities that they want to announce, please feel free to do so. If you're looking for collaborations, that's also fine. We'd appreciate that as well. We will not have a steering committee meeting this week. We will have one next week, Friday, September 15th at 11 a.m. EDT. And then again on the following weekend on Friday, September 22nd at 11 a.m. to get back in phase with the seminar series. Next week, we're hoping to have an industry representative come to the steering committee and talk about industry interest and concerns related to what we're doing in this working group. As far as our ongoing seminar schedule goes, we have on September 21st, Francois Grenier, who is going to be talking about the interesting and highly controversial topic about where SARS-CoV-2 came from. He's a biophysicist who's worked on a whole variety of interesting problems over the years. This one, he doesn't shy away from controversy. That one will be quite interesting. We have talks from University of Cincinnati and City University of New York. We've heard it from Bruce Lee before. We also have an industrial speaker coming up on November 16th. We have still openings for November 2nd and December. If you have suggestions for speakers, please do make those suggestions. We would appreciate that. As always, you've heard me say it many times. You feel free to suggest yourself, even if you've spoken before. Your graduate students, your postdocs, this is an open forum. We also are happy to try to invite anyone that you would like to hear from, who you think would further the community or be interesting for the research you're doing. Lorenzo is extremely efficient at getting people to come to speak to us, and we couldn't do it without your input. So I don't want to take any more time from our speaker today. We're going to be hearing about very comprehensive modeling approaches to aerosols, and I will turn the screen share over to you.
Good afternoon. Thank you so much for giving me this opportunity to share my lab's research on using the multiscale CFPD-PBPK TK-PD model to predict the health endpoints for respirable aerosol in human respiratory system and the whole human body. So my name is Feng. I am an associate professor in chemical engineering at Oklahoma State University in Stillwater. So first, I would like to thank our working group to invite me to give this seminar. I would like to also thank our academic partners, ANSYS and ESSS, to provide their computational resources to my lab to facilitate our research. I would also like to thank all my graduate students and funding agencies to make our research progress become possible in the last couple of years. Last but not least, I would like to thank all the pioneers, professors who actually working in the computational long aerosol dynamics and who also helped my lab to shape our research a lot in the last couple of years. Thank you so much. So today, I would like to first talk about this CFPD-PBPK TK-PD model, like why we need to use that, what can we generate using that multiscale model, and how to run this kind of model. Then I would like to go into a little bit more about what we did in the last couple of years to develop the next generation lung model. But again, our focus is about the pandemics, about the SARS-CoV-2 virus-laden droplets, transmission, transport, deposition, as well as the trigger immune system response. So one example we would like to share with you is how we bring the simulation to health endpoints by simulate different scenarios for SARS-CoV-2 exposure and also characterize the delivery dose in the lung and then the after-deposition dynamics. So what we are doing using this multiscale model in my lab. So everything started from the computational fluid particle dynamics. As you can see from the animation we showed here, you can use this kind of CFPD model to simulate the inhaled airflow, inhaled gas and vapor, as well as the inhaled particulate phase, and track their spatial temporal distributions of the concentrations and the particle deposition locations. And we also have this kind of what we call the elastic lung model, as you may show in the right. I will talk a little bit more about this. So we cared about, like what I said, it's about the spatial temporal distribution of all the variables of interest in the lung airway system. But we also understand that the deposition and absorption of the inhaled aerosols is not the end fate of those kind of chemicals. So we further view the interconnection and also the PBPK, TK, and TD model so that we can obtain the subject-specific deposition and absorption and converted that into the initial conditions for the other side, which is a set of the ordinary differential equations to either track the plasma concentration and other concentrations in other organs over time, or track the cell count over time for different type of those cells. So over the pandemic, we built this what we call the virtual human system. So as you may see, that virtual humans is for different genders, different ages, and for each virtual human model, we have the lung airway connected at the mouth and nostrils with those body shells. So we use this kind of virtual human and put that into different indoor environments to investigate the airborne transmission and determine the exposure level at the head region of some of those dummies inside of different conditions. And talking about different respiratory diseases, this is our motivation. So we are not only motivated by solving problems like related to lung cancer, but also we want to think about anything related to chronic lung diseases. So the reason we think that CFPD and PBPK TKPD model can be helpful beyond, or let's say, be complementary to existing in vitro and in vivo methods is especially about its special capability to provide high resolution spatial temporal distribution of variables of interest. Usually those, well, based on my biased understanding, those information are sometimes hard to be measured using experiments. For example, if we want to know that how we can deliver drugs into the small airways to treat asthma and COPD, or we want to target the tumors by deliver the chemotherapeutic particles directly to the tumor site, or we want to learn that how the virus deposit in the lung airway and trigger the immune system responses. The most interesting information we want to grab from the simulation is the local deposition of the inhaled aerosols. So this is exactly the reason why we want to use this multi-scale model to learn more about this whole aerosol transport process and unveil more underlying mechanisms. Talking about more details about this model. So I want to briefly show the modeling framework about this multi-scale tool. So using the SARS-CoV-2 as an example, we always want to start to model the emission and then their transmission in the indoor or outdoor environments so that we can determine the exposure level to certain human. Then with that exposure level, we can use that as a boundary condition, goes into the lung airway model to determine the delivered dose of different type of aerosols. For this case, it's SARS-CoV-2. And when we know the virus load in the lung airway, then we converted that and connect that information into the what we call whole cell dynamics to predict different type of cell counts that can represent different mechanisms that's triggered by those virus infection. Another example is not about the virus, but about the pharmaceutical application. So the modeling framework is more or less the same. If we want to determine the performance of a certain inhaler, we always want to start from the left, which is step one, to learn what kind of aerosol transport and interaction mechanism will happen inside of the inhaler flow channel, so that we will know that the particle size distribution and mass coming out from the inhaler. And that is also can be applied as a boundary condition into the lung airway simulation, and we determine the delivered dose, and then determine the chemical concentration in different organs. So all those kind of information can be used as quantitative evidence to evaluate the performance of certain inhalers. So when we think about the whole flow, we always start from those 3D airway reconstruction. It is started from the medical images, but unfortunately, sometimes the resolution of medical images cannot support us to generate a whole lung model with those small airways. So sometimes we need to create those small airways by just artificially generate some cylindrical pipes and connect it with the upper airway, which usually can be obtained from the medical images. And then when we have the geometry, we started to generate the computational mesh. And definitely model validation and optimizations are very important. And in my lab, we usually do those 5 different categories for validation, but due to the time limit, I save that into the supplementary slides. So if you guys have any questions, I can show more details about our validation process. So that step 3 is definitely just iteratively got involved with the solving process. When we try to get the geometry and the mesh ready, then we started to include all those conservation laws, which are the first principles about the CFPD model, so that we can predict the aerosol transport in the lung airway. And after we got the deposition and absorption of those inhaled aerosols, as I mentioned, it will be connected with the PBPK, TK, or PD model. So for the multi-phase flow model, we use to predict different aerosols. This slide shows the governing equations to predict the airflow and the discrete particulate phases. And this slide shows the airflow and the inhaled gas or vapors. sometimes we need to consider the what we call advanced mechanisms between those particles. For example, if we think about dry powders or droplets, we need to think about how those droplets or particles will interact with each other. So they may have a coagulation, they may have agglomeration, deagglomeration. So in this case, we need to employ what we call the discrete element method to simulate the adhesive and the repulsive forces between the particles or droplets. And we also use this discrete element method to simulate flexible fibers like nasal hair and cilium. And this is a very busy slide, but I just want to briefly bring this up at the very beginning of the presentation. So this is one of our host cell dynamics model, a set of ordinary differential equations. So the variables in each equation is the cell counts for different type of cells. And talking about the PBPK or TK, this is how we convert the deposition and absorption of those aerosols in the lung to an initial condition, which can go into that ordinary differential equation system so that we can track the chemical concentrations in different tissue groups. And sometimes we need to zoom in and model more explicitly about interconnection between the airflow side, the airway side and the blood flow side. So usually we do 3 layers, as you may see to the right of this slide. So it's like airway lumen, epitheliums, and subepitheliums, and goes into the blood circulation. And each layer will be governed by an ordinary differential equation, and the variable we want to solve is still the concentration. And in those equations, you may see there are the ADME, different mechanisms can be involved. So that's mostly about our equation system. And if you would like to learn, not learn more, but to know more about those equations, you're welcome to check our tutorial paper, which was published in 2021. So the long-term goal of my lab is to build what we call the next generation lung model. And one of the most aspects we think about is how we can make this virtual lung model be more physiologically realistic. So any neglected mechanisms that can influence the air particle flow dynamics prediction need to be considered and can be considered. I will briefly show a couple of examples, which basically that bullet is not our main focus today. And as I mentioned before this slide, we also try to just model the advanced particle interaction and transport dynamics, like droplet size change due to the evaporation condensation, particle-particle interactions, and even non-spherical particles. The third bullet is to our focus, and I will show that example later. And we also think about how to integrate the advantage of artificial intelligence into the kind of research. So we can use the CFD and deep learning together to maybe just develop some prototype of the novel diagnostic method or maybe novel treatment. And also thinking about inter-subject and inter-species variability, definitely that's something that we need to address, and I will show briefly in the next couple of slides. So the elastic lung model is one of the things we try to push through to make sure that our virtual lung model is more realistic. Usually, existing lung models, they are just assume that all airways are static. So the deformation kinematics was not considered. This is something that we want to just develop and put that into our simulation. And there's one example later, and I can show more details about how we achieve this using the dynamic mesh method under CFD. And this is our most recently work, which is how we just try to investigate the influence of the nasal hair on the inhaled airflow and particle transport in the nasal cavity. Before this study, to the best of our knowledge, all these kind of simulations neglected the presence of nasal hairs. But if you would like to read more about the details of our findings, you can check that paper I listed below. So what we found is that nasal hairs can regulate the inhaled airflow, make it more evenly distributed into the nasal cavity. And then the nasal hair, as you may see here, they can also trap a lot of particles. So if we neglect the presence of nasal hair, then they give us a biased prediction about the deposition of those particles inside of our nasal cavity. And we also do some of those kind of simulations, and you already see this. So we use this kind of modeling framework to help one of the pharmaceutical companies to evaluate their generic inhaler design compared to the hand inhaler, which is very popular in the market. And talking about health endpoints, those kind of simulations, we did investigations about the medical use of cannabis. We did the Influenza A virus simulations. Also, we did the xenon gas as the initial gas to see that what kind of the delivered dose into the blood circulation system. And also, we simulated the nicotine and acrolein coming out from the e-cigarette and track that blood concentrations, not only the deposition, but the concentration in the blood. And talking about this interspecies study, the motivation we have is that we learned there's a lot of animal studies. There's a lot of animal studies measure certain quantitative evidence or data using the animal, and they would like to scale that data up by timing the body mass ratio or timing the lung surface area ratio between the animal and the human, so that they can just scale it up and project the human response or maybe the deposition in the human body, which we feel that is not 100% the right way to do that. Because as you may see in this slides, that the rat and human or even other animals, their lung airway or the respiratory system anatomy is quite different. So the deposition in animal and in human lungs should not be proportional to their body weight ratio or the lung surface area ratio. So what we did is that we utilized that chance to simulate the deposition in the lung for different inhaled aerosols and in the mouth airways. And we know the deposition masses in the two species, so that under different conditions, different particle size, different inhalation flow rate, we generate a correlation, so that using that correlation, when people got some animal study or let's say a rat study data of the deposition, they may have a more, let's say, more accurate scale up factor that they can use to just to project what actually will goes on in human body, rather than just use some simplified scale up factor. We also noticed that most of the CFD studies, but this is like a couple years ago, usually people use one 3D airway geometry. But as you may see in this slides, the upper airway shapes could be very different from human to human. So what we did is that we changed the upper airways, and then we keep the tracheal bronchial trees the same in our 7 virtual model. So we try to see how that upper airway anatomy can influence the airflow. As you may see, there's a lot of differences, especially about the jet core generated in the trachea. And also, we see that how that kind of anatomy can influence the particle deposition in the lung airway, especially in the tracheal bronchial trees. So we did find significant difference. So what we want to do is try to claim that this is important. So we need to sometimes think about the subject variability in our simulation. So talking about AI-assisted diagnostic method or treatment, one of the things we achieved is we developed a prototype using a deep learning method. That deep learning algorithm can do something like this. So if the patient can be scanned, and when they do the exhalation, they have a airflow velocity contour in the middle of their chest. That contour is like the input into the convolutional neural networks. So the output is the algorithm can tell us where is the obstruction in small airways. So we created like 990 different obstruction cases and generate all those CFD data, label them with their obstruction locations, and train and test the 2 open source CNN algorithms. The outcome turns out to be really great. So the average precision is like over 93%. But we understand that is because our CFD data is so clean. So in high resolution, there's no noises. So if we replace those CFD data using medical images, the precision will become lower, but at least I think that shows a feasibility to use this kind of AI model to invent some of those maybe new non-invasive early detection method. So now I would like to go into a little bit details related to the topic of pandemics. So what we want to show you is about this whole framework, how we want to model the airborne transmission first, and then the long deposition, and then immune system response. So there are three kind of examples, which are not in a single project, but they represent those three different steps. So the first example I want to show is about how we predict the emission and transmission and lead to some mitigation plans, or maybe to understand more about how we can optimize mitigation plans for the infection risk due to the airborne transmission of those viruses. So what we did is that we have the virtual human. So at the very beginning, the first paper we published is about how the 6 feet social distancing policy makes or make no sense, or maybe can be improved. So as you may notice that at the very beginning of the COVID, we used that 6 feet social distancing policy. But we looked into more details about this policy. It is based on some assumptions saying that the two persons stand against each other and there's no ambient wind. So the static air is surrounding them, and there's no difference between different humidities in the air. So that bring to us a question that if we have ambient wind, especially like we live in Oklahoma, so there's always big wind, and also the humidity is changed from day to day, will that 6 feet separation be still sufficient to protect people? So this is the kind of the virtual model we built. Like two people stand against each other. The left guy cough out some virus-laden droplets. The right guy, basically there's a kind of like a guy just was subject to the exposure of those viruses, possibly. And we use the cough waveform and droplet size distribution as shown in this slides. So we run airflow with the ambient wind coming from different directions, and this is the wind coming from the back of the left guy. So we can visualize the airflow field inside of this virtual chamber, and you may see some circulations, and you may see some of those special wake flows in between those two humans. That is very important to help us to understand later on how the aerosol transport from the left to the right. And in this slide, basically those aerosols have different sizes, and they follow the airflow goes from left to right. So you may see some large red droplets, and those are exaggerated in size, so nobody cough out a basketball or like a soccer ball. It's just like 2,000 micron. And you see that those particles followed by like can sediment really fast because of the gravity, and this cannot reach the head region of the guy standing on the right. But if you see those small droplets, they actually can follow the airflow really well, and they are influenced by the turbulence. They can suspend in air much longer, and they can transport much farther than 6 feet. So that just gives us an idea that saying that 6 feet may not be 100% protected if there is an ambient wind. And we run different humidities, we run different wind conditions, and there's more details you can see from this slides. But due to the time limit, I will just try to skip this part. But also, there's one thing I want to bring it up is like that's kind of the beauty of using models. So we can play with the boundary conditions virtually. So we are not only simulating steady state wind, we also simulate the gust. So it's like gusts come in, and then the air becomes static, and then the gusts come in again. So that kind of gust wind actually break down the wake flow, like the recirculation will not exist between those two humans. So what that caused? That caused the droplets or the aerosols transport even faster from the left guy to the right guy, because no droplets will take the recirculation motion. It's always just go forward. It's actually just give us some new idea about the complexity that the wind condition can can generate to increase the infection risk to the other guys. We also use the same model to evaluate a air sanitizer, and we put that into a patient room. So this is something motivated by industry company, and they have a design. They want us to test that for them. So that what we did is like we follow the CDC and OSHA's recommendation as when people think about mitigation plan, the top option is that you can eliminate or remove all the pathogens. So this is the design provided, and what we did is that we create a virtual patient room, and that guy lying there is actually got infected, and the droplets coming out from his or her mouth. So we want to play with the ventilation conditions, and also want to play with the air sanitizers flow conditions. We want to make sure that this sanitizer is very robust. So no matter what kind of ventilation conditions there will be, the presence of that air sanitizer will still reduce the suspending virus concentration, which make a positive impact to reduce the infection risk of any people goes into that room. And this is the numerical setup we did. And we did run 3 different ventilation conditions, and for the airflow through coming out from the basically the operational conditions for the air sanitizer, we have different flow rates running through it. I will skip those common equations, but if you have any questions, feel free to ask me after the presentation. So same thing, we can visualize different size of those droplets, and all those kind of droplet dispersion through different cases. There's a lot of animations in that paper. But one thing I want to stress is when we check about the suspending, like the figure B, when we check about a suspending droplet number, which somehow related to the virus concentration suspending in the room with different ventilation conditions. As you may see, that with ventilation condition B, if we turn off the air sanitizer, the suspension is actually lower than the case that we turn on the sanitizer. So that gives us a clue to say that it's not like we have an air sanitizer, then we put that into a patient room. It will always help us to mitigate the infection, the infection risk. Indeed, it needs to be well coordinated with the ventilation conditions. And also for the design of those air cleaner or air sanitizer, they need to think about this, and they need to make sure they test different ventilation conditions and make sure their air sanitizer can work properly, and they may have a more solid design, a more robust design that can do this no matter what kind of ventilation condition that would be. We also simulated the kind of the emission of the virus in the classroom at OSU. As you may see, the guy got infected is circled by the red square. And you can also see that large droplets basically falls down under 6 feet. So one thing is that all the virtual humans are separated with at least for 6 feet. But you see the small droplets, same pattern. They basically can follow the turbulence in the airflow due to the ventilation, and this can fly really much farther than 6 feet. And this is another angle. And as you may see to the left, to the right, that's the deposition pattern of those small droplets. Can even reach the podium. So it's pretty impressive when we saw this kind of simulation results. So in that case, we tested about the virtual mask. For your information, that virtual mask is not just have a good seal with that virtual human face. There's a 1 centimeter gap. So we exaggerated that gap with 1 centimeter. But still, if you see that kind of results in deposition, there's no small droplets can travel further than a circle with a 6 feet radius. So that's just somehow shows physically that mask is relatively helpful. Those are how the models can work, can provide the results to help us to determine the exposure levels at different locations inside of different indoor environments. So when we got the head exposure levels for virtual human. Then we can start to think about apply that concentration as the boundary condition goes into our virtual lung system. So for the case I want to show is the simulations we did using our elastic whole lung models. So as I mentioned at the beginning of this presentation, the human airway has a lot of motions during the inhalation and exhalation. It's like our vocal fold is always open and closed, and airway is always expanding, contracting. And also due to the medical image resolution problem, the lower airway sometimes is just missing from those 3D virtual lung models. So what we did is trying to find a way that we can create a whole lung model, as you may see, that we do have a truncated airways, but it's coupled on the boundary conditions. So in each lobe, there's a one branch all the way goes down to the alveoli, which the alveoli is a heterogeneous pars media model that you can see to the right. What we actually achieved in this model is the glottis motion and also the airway expansion and contraction during the prediction of those transport and deposition of inhaled particles. And the first thing we did is that how we can model the glottis motion and with validated kinematics. So what we do is that we use the dynamic mesh method. As you may see that there are some governing equations here. So those equations help us to prescribe or predefine the nodal displacement of the mesh of all the mesh nodes inside of this orange area, orange region, which is our glottis. And using this set of a generalized equation, actually the glottis motion predicted by our dynamic mesh method can fit clinical data very well. But also we do not want this kind of governing equations to be limited to just one glottis motion. We want it to be more generalized. So what we do is that we revise it and try to make it like a general set of equations. So if you see the equations in this rectangle, the fx is adjustable, gt is adjustable, and in fx, that exponent is adjustable. So all those kind of parameters can be changed, can be varied. So we just using this kind of general equations, you may see that we simulated different conditions, and you see that at the bottom of this slide, there are different set of clinical data represent different patients, and the aperture opening from time to time can fit experimental data pretty well. But after we achieved that glottis motion prediction, we started to think about how we can model the airway deformation. Airway deformation is anisotropic rather than isotropic. So one thing is that because the presence of our rib cage from the left to right, the deformation of airway is quite limited. It's just like 1/3 compared to from head to foot or from the back to front. So with the support of the pulmonary function test results, we compare our numerical model with the experimental data in the lung volume changes, not only for healthy human, but also for COPD patients with different GOLD stages. So this is actually the final results and the final equation we use. It's use the same strategy that we just use this kind of nodal displacement control from time to time to get this airway deformation from time to time. And one thing is that elastic airway model will significantly increase the computational time. So we do want to investigate it and see that whether it is worthwhile to include those elastic model in those simulations. So the things we compare, like what you can see from this slide, that we use the static long airway model and the elastic long airway model to predict the particle depositions in the whole pulmonary route. And we can visually see the big difference. So that just gives an idea that the elastic airway model needs to be included sometimes to make sure that the prediction of the particle deposition is not biased in the airway. We have other results, and you can find them in the paper I cited in this presentation. So after we got the lung deposition or the delivered dose in the lung, we started to think about how we can just move forward and maybe simulate a little bit more to the endpoints. So the example I show here is also done by my also recent PhD student. And we try to simulate the nasal vaccine spray droplets inside of the nasal cavity, and after the droplets deposit in the nasal cavity, and what kind of trigger immune system response we can get. So we are motivated by the availability of intranasal vaccine to somehow work against COVID-19. We think it's important because some people may be afraid of needle, especially this is intranasal vaccine is very friendly to young kids. But there is a controversial results we found early last year. Some people say that intranasal vaccine is good, it's effective. Some people found it's not effective. So there are knowledge gaps opened. It's still question marks about all the things that are listed here. So we think about what is the possible reason for the ineffective immune system response led by using the intranasal vaccine. And is there anything we can do to improve the effectiveness? Maybe just give a little bit of targeted delivery strategy. So by saying that, we try to employ the composition of a vaccine that we found in open literature with 4 components, and there's a 1 component in the water, which is the evaporative components. We investigated in different spray cone angle, different spray droplet initial velocity, and different initial droplet composition, like the vaccine composition, and see how those factors can influence the delivery efficiency and maybe the virus titer change over time later. This is the airway model we used. It's a 6-year-old child, and this is from our collaborator, Dr. Froman. We are trying to target the olfactory region at the top of the nasal cavity, which is ACE2 reach. And all the spray nodal diameter, droplet size distribution are coming from the experimental data about the COVID-19 vaccine we investigated in this study. And as you may see here, that olfactory region is just a circle here on top of the nasal cavity. So I will skip the equations. These are the kind of the different compositions we use in this study. Also, the equation has been shown at the very beginning of this presentation. But one thing I want to talk about is about calibration for this whole cell dynamics model. As you may see, there are 30 ordinary differential equations here, and there are 50 coefficients. But unfortunately, not all those coefficients can be calculated or predicted using theoretical calculation. So what we did is that we tried our best last year to find some of the available experimental data, and we used a multi-objective genetic algorithm to do the optimization of those coefficient values. It turns out that the modern results shows reasonably agreement with experimental data we show here. So we started with the simulation, still simulate the airflow and the droplet transport in the nasal cavity. And what we found is that when we use different initial velocity of those spray, it seems like the deposition fraction inside of the olfactory region does not change too much. So this is because the spray velocity is very high. So the size change of those spray droplets does not influence too much about the different time they traveled from the emission to the olfactory region. But we do see that when we change the spraying cone angle, there's a big difference, no, not big difference, but there's a noticeable difference for the deposition fraction inside of the olfactory region. When we have a smaller cone angle for the spray, it actually lead to a higher deposition fraction in the olfactory region. So it's more like you have a narrow spray, so it can target the olfactory region better. But another thing I want to show is that although the deposition led by the 5-degree spray angle is higher, but you may see that the distribution of those deposited droplets not as spreaded as the 10-degree. So later on, we found out that when we have 5-degree as our spray angle, the coverage of those epitheliums inside of the olfactory region is not the highest. It's actually the 8-degree give us the highest coverage, vaccine coverage inside of the inside of olfactory region for those epitheliums. So we play with those things and connected those information, converted that into the input into our whole cell dynamics. And we hope that we can see some difference in the viral titer curves. But unfortunately, with all those different angles, spray angles, we do not see big difference in the viral titer curve or the time profile. So that is that leads us to think about, is that some of the ineffective nasal spray vaccine actually due to the small area that the spray can cover. So we just play with that vaccine coverage percentage, goes from 0% to 99%, and we started to see big difference. And the virus titer, like the peak, become different, and the decaying become like sometimes it's faster, sometimes it's just not that fast. So we think about like if there is any novel nasal spray strategy can be developed so that the spray can cover maybe a higher percentage of the epithelium inside of olfactory region, that may just give a really effective effective performance in the vaccination. So that is basically all about the example that I want to talk about today, to show how we try to use this multiscale modeling framework to bring the simulation to health endpoints. But there are other things that we can do to create a more physiologically realistic, elastic long models. And we would like to think about our future work so that we can make this model more realistic, and it could be someday can be used as an in silico clinical trial platform to either help on the pulmonary healthcare or maybe can help on the occupational exposure risk assessment. So yeah, that is all that I want to present today. Thank you so much, and I'm happy to answer any questions.
I'm wondering, so I come from a slightly the opposite side of things, from the subcellular level modeling. Do you see a path, and I guess maybe first need to be able to integrate your layers into that scale?
So when you mean scale,
subcellular, cellular interaction signaling networks and that sort of stuff.
Okay. So if I understand correctly, it's about that 3 layers of interconnections, right?
Mm-hmm.
Okay. Yeah. So sometimes I think that is necessary, because if we do not use that, so when we try to connect with between the CFPD model and the PBPK or TK model, we just need to introduce those bioavailability factor. So sometimes the bioavailability factor is accurate, sometimes it's probably not that accurate. So if we can explicitly model the interconnection, that may give us a better idea about how we can more scientifically determine the bioavailability factor value. So that's my understanding.
I wanted to sort of cycle back to part of your earlier presentations on the background. Have you started to consider using these types of models to look at the inhalation exposure to, say, heavy smokers or people with COPD or with asthma?
Oh yeah, thank you so much, Tom. So yes, so but we do have that plan, but we never run that simulations using patient lung conditions. But what we did is that we did run a lot of those simulations and published a couple papers on the inhalation of e-cigarette aerosols, and see that what kind of deposition we will get inside of the lung airway. Especially if the recent paper focused on the different nicotine forms, how those nicotine forms can influence the evaporation and condensation through those transport of those aerosols, and then influence the deposition. So yeah, we did investigate that, but before that, we did not think very much about the patient, because that's just one of the papers that we just it's more like the first paper we try to focus, goes back to what I did in my postdoc about e-cigarettes. But yes, so we using the elastic airway model, we should be able to at least reflect the disease-specific airway deformation, and that can, I believe, that can reflect more physiological characteristics of patients with different lung diseases, and that will lead to some difference in the deposition prediction that we can see. And also, not even to mention that the airway lumen will be different from healthy people compared to people who have lung diseases. I hope that answered the question.
Yeah, I guess the other question I had was, are you familiar with the lung-on-chip model from the Wyss Institute?
Oh, sorry, I did not quite catch it.
The lung-on-chip model, the lung-on-a-chip?
Oh, lung-on-chip. Yes, yes. I did not know the details, but I heard of that and I read a couple of kind of highlights in on LinkedIn. So not goes in. I did not get a chance to go into details about that. Yeah.
I guess what struck me is when you look at some of their simulations, they see that the expansion of the alveolar space really has a dynamic effect on local blood flow and macrophage transport. And it would be sort of cool to integrate some of that biology into your simulations.
Yeah, thank you so much. That's a great suggestion. So we will definitely just go into that, and that's also one of the limitations for now for our model, because we simulate aerodynamics, and kind of those how the kind of the airflow domain can be coupled with the blood flow domain. That's still, I think our model now is still quite the resolution is not that high, so we feel that is definitely something that we can improve. So make sure that there's more things we can explicitly model rather than just simplify that into maybe just OE or whatever. Yeah.
And then just one more question, if you don't mind.
Oh, go ahead. Yeah.
So at the EPA, I'm at the EPA, and there's a lot of interest in ozone. I'm not an ozone researcher. I'm an ozone wimp, but there is a woman at the EPA, Colette Miller, that's got a really interesting finding where she finds that if you expose pregnant rats to ozone, you get a cytokine storm in the lung, and that affects placental development only of male rat fetuses, not the female rat fetuses. So there's really some very interesting interactions between lung dynamics and placental dynamics during pregnancy for things like ozone, maybe other types of exposures.
Yeah, thank you so much. Oh, that's interesting. So yeah.
Okay, thank you.
Yeah, thank you so much.
Okay, other questions for our speaker?
Yeah, so I'll ask one. So my name is Mitchell. I'm a postdoc at UC Irvine. So it was a great talk. I'm kind of following up with the previous question, which is a good lead in. So I come from hemo modeling and simulation, but for the pulmonary circulation background. So you know, sort of similar to what you're saying. I mean, the next goal on my list is to look at, you know, multiscale linkage, but looking at alveolar interactions with the pulmonary capillary beds. But I was going to ask, is there any benefit in terms of like computational speed up to consider doing, instead of doing 3D airway dynamics, to maybe look more at, you know, I've seen one-dimensional theory where you're looking at like axial propagation. Do you miss out on a lot of the dynamics that you're interested in if you're doing 3D lung dynamics, or do you think you could get away doing 1D dynamics for airway dynamics and then linking that to your more compartmental models there of inflammation?
Yeah, thank you, Michel. That's a great question. Yes, so I think it depends on the regions you want to simulate. If it's let's say that if you're concerned about the upper airway and central lung, so with the airway, so those airways are with larger diameters and complex boundary shapes. 3D is so there will be a lot of what we call secondary flow. So it's like your airflow will be totally like it's totally like 3D airflow. So you may see a lot of circulation. You may see a lot of kind of uneven distribution of the airflow through the pipes in each cross sections. So in those regions, I believe 3D simulation is necessary, although it's costs a lot of more computational resources. But when we say that the flow goes into the small tubes, like the peripheral lung, the airway is small, the Reynolds number is lower, much slower. So it's more like the 1D model can really do a good job for small airways. I remember my student, like that Hamide did that rat simulation and human simulation. If I remember correctly, the flow field she saw in the simulation using that 3D model is that when the flow go beyond the generation 13, it's more like it's fully developed, like just like 1D flow. So if I can comment on this for now, based on what I learned from all my labs work and other people's work, for small airways, 1D model should be a good fit. For upper airway and central lung, I believe 3D model is still important, because if we simplify those flows as a 1D flow, we need to make some corrections on the particle deposition prediction using the 1D model. Yeah.
That's it. And then one follow-up would be when you're, and you might have mentioned this, I might have missed it, when you're linking the airflow dynamics, sort of particle deposition to the ODE level models, what are your opinions on accounting for sort of spatial interactions in the ODE scale, where, yeah, I don't know if you're accounting for that yet.
Yes. Yeah, actually, I did not talk about that, but that's a great question. Let me see if I can find some slides which can yeah, maybe these slides can support a little bit. If I understand your question correctly, it's more like regional effect, right? So different regions have different bioavailability factors, and we cannot just consider everything just as a whole piece, like all deposition as a whole piece. So we basically divided. So if you may see in this slides, so we divided the whole long model into different regions, and those different regions will have different, let's say if we do not explicitly model the interconnection part, we will have different bioavailability factors for different regions. So that's count for the difference between like in different sections of the airway. And when we add them up, so like the deposition is more like a sigma deposition, let's say deposition mass in certain region times bioavailability factor. So that sum will become the kind of an initial condition, goes into the ODE system, and then we can start to go calculating. So if we do not explicitly model the interconnection, that will be the things that we are that is the thing that we are doing now.
Okay, got it. Thank you.
Yeah, thank you so much.
There was a question in the chat about if you could elaborate a little bit more on how you use AI for model enhancement and data integration.
Sure. Thank you so much. I'll go to that slide, or this should be front. Sorry. For that, trying to find that slide. Yeah, it's here. So, so what we did is like this. So we got motivated about the this idea was motivated by, you see that virtual human and the airway in there. At the very beginning, we just simulated two cases. One case with all the airway are opened without any obstruction. Another case is that we just artificially block some of those airways. And we run the simulation as two acceleration, same boundary condition, just sorry, not same boundary condition, same flow rates, but different small airway openings, like one has some obstructions. And we observe there is actually the flow velocity difference that we can tell at the cross section. You may see there is small cross section. Let me see if I can do that in. So there's a small cross section here, and we just try to compare the velocity distribution on that 2D cross section in the trachea, and we see difference. So that just gives us an idea, but which is fluid dynamic is always true, that when you change something in the upstream, like the geometry, like the opening, the downstream velocity field will change. And we think about, okay, so that velocity profile inside of the trachea in the chest can be used maybe as an indicator to tell where is the obstruction. But at that time, we did not know if the deep learning network can do that. So, So what we do is that we create 990 different obstructions using the same airway geometry. You can think about that all the small airways are blocks of 1 or 2 for each cases, and that gives us a full combination of about 990 cases. And we run all the exhalation simulations, harvested 990 different velocity contours at the same cross section in the trachea, and all those contours are labeled by the obstruction location, and that sorry about that. And that those kind of labeled images is our training and testing data. And we use 80% of those for training, very classical, right? So 20% for testing. So that goes into the neural network system. Well, this kind of structure is not accurate. We use, so the two deep learning model we use is one of the Euro V4, and the other one is ResNet 50. So the output is like, the output is, I need to mention that, sorry, I skipped that too much. The output, we try to just do this preliminary study by creating 3 different classes as output. Class 1 is that we have obstruction in the left lung. Class 2 is that we have obstruction in the right lung, and class 3 is like we have obstructions in both lungs. So that is kind of the work we have done. Like you may see more details in that paper. I'm not sure if this explained more clearer about this whole process.
Okay, well, thank you. I had a question just about the complexity of these models. You have these very detailed, large-scale CFD models. You have the in-lung CFD models. You have the transport models across the lung epithelium. You have these detailed models in particular choke points. Are those models written as sub-models that are then interconnected with each other, or is this all written as one enormous single solved integrated monolithic model? In other words, you draw it here as a series of sub-models.
Yes.
But how do you actually describe and interconnect those sub-models? Because some of the coupling is quite tight, and that must be numerically quite challenging to be able to interconnect these models at different scales. I wondered if you could talk to us a little bit about that. Maybe we need to have you come back and give us a whole talk on multi-scale interconnectivity of complex models, because that's a topic of great personal interest to me. But please, if you could tell us a few words about that, I'd appreciate it.
Sure. Thank you, Dr. Glaser. So, the model, like the step 1 and step 2, they are coupled by the boundary condition we assigned at the mouth of either at the mouth opening or the nostrils. So step 1, we got the exposure level, let's say the concentration, or maybe the particle numbers. That how to say, the right side, that guy, that virtual guy that have added his mouth and nose, and those information is exactly the boundary condition can goes into the lung airway simulations. We did run one of those simulations for welding fume without using the boundary condition to couple them. So we have that virtual guy with the lung airway connected at the mouth and nose. That's for welding fume exposure, and we. So for that part, for that model, there's no need to connect those 2 modules. They're already together. From between step 2 and step 3 is actually just how it's just a matter of fact that how we can convert the deposition and absorption, either the mass flow rate, let's say the not mass flow, sorry, it's the mass flow rate or the total mass, and converted that into something we can input into the step 3. Either PKPD or maybe the TK model as the initial conditions goes in there. So I agree with you. The time scale is different. So for the inhalation, the scale is very small. So there is an assumption saying that if I inhale something, I got a lung deposition. It is even I have, let's say I use e-cigarette. I have 10 puffs, and we we grab that total deposition values and just combine them together as one initial condition for the ODE system at time 0, because the temp-ups is still very short compared to kind of the PK time scale. It's maybe hours or maybe days. So we just make that assumption. I did have one student when he simulated cannabis, he did simulate it like kind of transit coupling, like inhale, like deposition goes in, it's more like signals goes in, and different initial condition at different time goes into the ODE system. But later on, we see that it's not very necessary for us to do that. Yeah. So
the separation of timescales means basically that you could run the models independently and treat them as boundary conditions for each other. In this case,
yes,
now,
yes.
now in the lung inhalation model, where you're doing the biomechanics of the lung, then you can't separate that from the fluid flow.
Yes, yes.
And then you also had a transport across the epithelium as a submodel. So there you had tightly coupled models that are operating at the same time scale.
Right. So the interconnection, that explicit interconnection layers, they should be connected with the with step 2. They should be tightly coupled. Basically, their time scale cannot be. You cannot assume that time scale is different. It's quite similar. And for if I can provide more information about interconnection layer, so that small module was written by what we call the user-defined functions. So in C program, so that is just for the lung airway simulation is more like I cannot say it's like a post-processing code that you run, but it's more like an additional layer for the calculation, but based on the instantaneous deposition pattern, so that it is very tightly coupled with the CFD module we use. Yeah, it's more like a little bit more customization. Yeah.
So may I follow up one question on that? And I don't want to take away time if other people have questions they want to ask, which is there within the field that you work in some kind of standardization so that, for example, if somebody else had a model of the airborne transmission, you would be able to interface that model with your lung deposition model? In other words, is there an agreement that there's going to be a description, a sort of what I would call a constitutive relation, although that really is talking about the solid or the fluid, but something like a constitutive relation or a computer science way of API, which says that we're going to agree that there's going to be a model of a human with a mouth and a nose, and there's going to be an inhalation, some kind of description of the inhalation that happens in that. Is there some kind of high-level description of these components that's agreed upon so that, say, you and Chantal could connect your work together? Or do you have to hard code that? At the moment, that would be done basically at C++ level.
Thank you so much for that question. For that question, I do not have a very clear answer for now. So I need to read more about that. But I believe it's achievable. But so what's in my mind is like, especially for the CFD part, like this part, if we want to make that connection really easier, I think the intersubject variability needs to be addressed well. So there should be a lot of different airway models that we use, not airway models, or airway geometries that we use. So large amount of data sets, and definitely in that case, machine learning and deep learning would help. So I usually feel that if we just every time when we got a condition and we run the CFD, it's very slow. It's a physics-based model, which is good, but it's just cannot provide instantaneous feedback and answers.
Right. I guess I'm trying to understand how much somebody else could generalize what you're using. In other words, the C++ code, the methodologies are already very impressive. But if they're wrapped in a way, for example, suppose that I had a mouse model of the lung architecture, and I wanted to take everything you did and apply it to the mouse. Well, I'd pull in the triangulation and the description of the mouse lung, but what would I have to do to be able to make a murine version of this model, for example, or go the other direction? I think Tom Knudsen may have been thinking about that kind of question a little bit. in terms of what it would take to make these models more componentizable, so that they were more reusable or more generalizable. They're incredibly impressive, but one can imagine. We've had talks, for example, from Nova Discovery, that's a virtual clinical trials company, and they have a framework architecture where you can plug in components like these. So you could, for example, they do clinical virtual simulations of clinical trials of airborne disease. And so they have a framework where, if, in principle, a model like this could be included inside of their framework to model the individuals in the trial. But you'd have to have some particular interface that connected your models to their components.
Oh, thank you. I think I understand better about your suggestions now. I think it should be making this kind of model should be more like more modularized, so that it can be just connected and embedded into other people's modeling framework. Like, yeah. So it's more, yeah, okay. So I think I got that now. Yeah, thank you so much. I think it's about what kind of data that we need to exchange between this model and the rest of the modeling framework. So I think that's a great idea, but I do not have I do not have a clear mind to how to do that now. So.
That's fine. I wasn't expecting that you would do it yourself. I just wondered, within the field at large, if that was something that was being developed to assist with the development of more interconnected models. I know that's a topic of interest for this particular community, which is why I brought it up. Okay, are there any other questions? We've had a good discussion. There's time for one more question if anyone has one.
Yeah, I have one.
Please, Idamar, go ahead.
Yeah, part of my chat was asking not only about AI, but also about big data analytics, which, if I look at the respiratory expert, they see different kind of patients. And maybe one could group them and find some characteristics of this group, and each group can be now tested in your model, especially.
Oh yeah, Itamar, thank you for your suggestion. We are indeed collaborating with another faculty at OSU and try to do something about that. I can only say that we are trying. So the things we are trying is that we have kind of medical images, like good data sets for COPD patients at growth stage 4. And my collaborator is an expert in AI. So what they want to do is trying to, well, hopefully I did not explain what they are trying to do in the wrong way, but they are trying to use the GAN algorithm to try to analyze medical images and grab the feature and to try to use it. That called data augmentation. I'm not sure, but they are trying to find a they can create a representative set of medical images that can be representative to that age group and that disease group. And what we want to do is that we grab those set of let's say the fake images, but it's representative, so that we can create a representative 3D airway model for that specific disease stages. So yeah, I'm not sure, but I think I talked too much. Yeah, not sure. Hopefully I explained their work clearly. I'm definitely not an expert in that field.
For example, heavy smoker is different than apnea patient, right?
Yes.
Okay.
Okay, wonderful. Thank you for the intensive discussion. Are there any additional questions? Okay, well, I want to thank our speaker for a wonderful presentation. It was really exciting to see this work. I'm looking forward to seeing how it develops further. I hope you'll come back maybe and give us some more technical presentation on some of the methodologies. You could really give a whole series of lectures on this work. There's so much here.